part 4 of seed technology basics discussion. Let us see seed production in different crops and some of the basics. First of all, the difference between hybrid seed production and varietal seed production. Under varietal seed production, it's a single parent multiplication, whereas hybrid seed production needs two or many parents, like you may be knowing about double hybrids, then double cross hybrids, triple cross hybrids, all those things. So there will be multiple lines were also crossed in order to get the hybrids. So in varietal seed production, isolation distance is less, whereas under hybrid seed production, it is more. Production is by open pollination under varietal seed production, and controlled pollination is practiced under hybrid seed production. Seeds can be used 3 to 5 generations, even many generations under varietal seed production, whereas every year we should change the seed under hybrid seed production. National Seed Policy 2002. Before this, the Government of India had adopted a new policy on seed development in 1988 to remove roadblocks in import of horticultural seeds and allow import of seeds of cereals and pulses in limited quantity. First State Seed Corporation was established in Pantnagar. Then National Commission on Agriculture's Seed Group was established in 1972. Technology Mission on Oilseeds and Pulses established in 1986. Now it is renamed as Integrated Scheme of Oilseeds, Pulses, Oil Palm and Maize, also called as ISOPALM. That means the 1986 Technology Mission TMOP is renamed as ISOPALM now. Seed Transport Subsidy Scheme was introduced in 1987. New policy on seed development in 1988. Then Seed Bank Scheme in 2000. Seed Bill in 2002, National Seed Plan in 2005, National Food Security Mission in 2007, Rashtriya Krishi Vikas Yojana in the same year 2007, Indian Minimum Seed Certification Standards 1988. So you know all everything in every of our video we have discussed a breeder foundation and certified, but nowhere I have told the definition of this. Just to refresh your knowledge, breeder seed is the progeny of nucleus seed of variety and produced by breeder. This is mandate of ICAR through ARS, SAUs, NGOs, etc. Foundation seed is the progeny of breeder seed. It can be produced from breeder seed or foundation seed. This is done by NSC, State Farm Corporation of India, then state uh, these universities and private agencies. Certified seed is the progeny of foundation seed. This production is responsibility of state government. Coming to varietal registration. Each variety has to pass through three phases of evaluation. First one is initial yield evaluation trial, also called as IET or preliminary yield trial. In different crops it is called with a different name. Generally it is called as preliminary yield trial. The second one is AVT, Advanced Varietal Trial or Uniform Regional Trial. You may get confused with these terminologies. You should always know the synonyms, where the preliminary yield trial is also called as initial yield evaluation trial and advanced varietal trial is called as uniform regional trial, or it is also called as coordinated varietal trials. The third phase is URT, from entries selected from phase 2, that is uniform regional trial taken from phase 2. Under PPVFR, that is Plant Varietal Protection and Farmers Right Act 2001, certificate of registration shall be valid for 18 years in trees and vines and it is 15 years for other crops. Seed certification is generally carried out in 6 phases. Indian seed industry is 8th largest in the world and annual growth rate is about 12 to 13%. So as per EPA 1986, all GM crops should be passed under Environmental and Biosafety Act. Okay, biosafety standards they should maintain. DSC is in consultation with ICR will prepare national seed map. In 1939, Association of Official Analysts AOSA was established. Sand is used in germination test of 0.05 to 0.8 mm of size. Germination paper used to be 46 into 29 centimeter, that is a standard size of it, and pH maintained is 6 to 7.5. 100% cellulose material is used here. Now, genetic purity maintenance in hybrid seeds. First one is nucleus seed of inbred lines maintained by self-pollination or sip pollination. Here sibling maintains the vigor. In nucleus seed of non-inbred lines, by giving isolation, roguing and other processes, and these are purified by mass selection. Remember, in non-inbred lines, mass selection is used in order to purify the lines. Coming to genetic purity, breeder or nucleus seed is always having 100% of genetic purity. Foundation 99.5% or 99%. Certified seed 98 or 99%. Quality of seed alone accounts for 10 to 15% of increase in productivity. Seeds of GM crops or genetically modified crops will be imported only through NBPGR, not through other sources, as per Environmental Protection Act 1986. Coming to the physical purity for certification, for maize and bhindi 99% purity, most of the crops 98%, carrot 95, sesame, soybean and jute 97, groundnut 96%. Coming to the objectionable weeds of different crops, in Barsim, shikori is the objectionable weed, cucurbits, wild cucurbit, kasuri methi, Melilotus species, lettuce, wild lettuce or Lactuca sativa, in bhindi, wild bhindi, that is Abel moisture species, in rapeseed and mustard, Argemone mexicana, in wheat, Convolvulus arvensis, in paddy, wild paddy or Oryza sativa variety, Fatua. Optimum moisture content for seeds for storage, for long term storage it is 6 to 8%, and for short term it is 10 to 13%. The formula for seed replacement ratio is x by y into 100, whereas x is the quantity of actual seed sown and y is quantity of certified seeds required for entire area. Seed replacement rate in India is usually 15 to 20. Now it increased up to 30% and it is 100% for hybrid seeds. Now, 3 routine tests in seed testing laboratory. The first test is germination test. Total 400 seeds in 4 replications, 100 seeds per replication are kept. A representative sample from seed lot is taken, that is primary sample. Bulking of primary sample is composite sample. Fraction of composite sample sent for testing is submitted sample. These terminologies are very important. See under germination test. Primary 4 replications, 400 seeds are taken, 100 seeds are kept per replication. Okay. Primary seed is representative sample from seed lot. Okay. Composite sample is bulking of primary sample. Submitted sample is fraction of composite sample sent for testing. Okay, in germination chamber where we keep the seeds for germination, we should maintain 25 plus or minus 1 degree Celsius temperature and more than 85% of the humidity. Seed moisture test. Here optimum moisture test level for cereal should be 10 to 12%, pulses and oil seeds 8 to 10%, and vegetables 6 to 8%. Coming to the quality control and agency of different classes of seeds, for nucleus seed, breeder or sponsoring institute will be the nodal agency, and quality control is through maintenance breeding. For breeder seed, breeder or institution are responsible agency. Field inspections are done for quality control. Foundation seed stage 1 and stage 2. Here, state department, NSC, SSC, private sectors, universities, all these people will take the act as agency, and state seed certification agency and field inspectors play important role in quality control. Then certified seeds given to farmer, and farmers can again multiply or make a cross with other seeds, and they can even sell it as truthfully labeled seeds. Here onwards it is farmers responsibility. Coming to seed production in different crops, first one is maize. Maize is Zea maize. The chromosome number is 20, 2n in diploid form. Inflorescence, panicle or cob. Flower, female is cob axillary, male is tassel. Pollination is cross pollination, it is protandry condition, means androecium matures first, then gynoecium. Flowering pattern, top to bottom, that is tassel, basipetal, bottom to top, or that is cob or female part, acropetal. This you should remember. In case of male part it is basipetal, and in case of female it is acropetal. Acropetal is bottom to top, means upside. Fertilization in case of maize, pollen shed at 1 to 3 days before silk emerges. Silk will be pinkish, receptive, turns brown after fertilization. Silk receptivity is 8 to 10 days, whereas duration of flowering is 2 to 14 days. Open pollinated varieties. Raise varieties under isolation of 400 meter in foundation seed stage, and for 200 meter in case of certified seed stage, and allow for open pollination. Here, along with hybrids, synthetics and composites play important role. Synthetics are variety obtained by mating in all possible combinations. A number of lines, that is more than 5, to combine well with each other are taken here. Example, COBC1. Composite produced by open pollination among number of outstanding strains. Example, K1, Jawahar, Vikram, Sona, CO1. Varieties. In varieties, isolation distance is 400 and 200 for foundation and certified seed. Seed treatment with azospirillum at the rate 50 grams per kg of seed to fix nitrogen. Seed rate of 20 kg per hectare. Then recommended fertilizer dose 150, 75, 75 kg per hectare. 2% DAP spray at 50% flowering stage for uniform flowering, and 20 kg zinc sulfate per hectare is recommended agronomic practice. Weed control with atrazine at the rate of 500 grams per hectare as a pre-emergency spray. Physiological maturity with cob sheath turn yellowish color. Formation of drunken layer or shrunken layer we may call. Then moisture of seed is 35% when it attains the physiological maturity. Xenia effect is found in maize, effect of foreign pollen on same generation. Then metaxenia is effect of foreign pollen in next generation or on the maternal tissue. Germination standards are 90%. Common varietal seed production technology in maize is open pollination under isolation. African tall is a fodder maize variety. Coming to hybrid seed production in maize, by detasseling, seed rate in female is 7 to 10 kg per hectare, male 3 to 4 kg per hectare. 4 to 7 to 8 border rows are maintained, generally 4 rows. Planting ratio 4 is to 2. In double cross hybrids and triple cross hybrids 6 is to 2, whereas for single cross hybrids 4 is to 2. Fertilizer dose for hybrid seed production 200, 100, 100 NPK kg per hectare. Number of inspections 4. 1 before flowering, 3 during flowering. Next crop is paddy. Here Oryza sativa is the scientific name. 2n equal to 24, diploid chromosome number. Inflorescence is panicle. Flowers remain open for 10 minutes and then closed. Isolation 3 minutes. To break the dormancy, soak in KNO3 with 0.5% for 16 hours or 0.1 normal HNO3 for 16 hours. Pre-germination is followed in paddy. Seed hardening with 1% KCl and pelleting with azospirillum with 600 grams per hectare. During nursery production, for raising 1 hectare of paddy, 10%, that is 800 meters square nursery is needed. Nutrients used here are basal dose of 40 kg DAP before pulling out of the seedlings. Then weeding with butachlor 2 ml per hectare or thiobendicarb 2 liter per 2 ml per liter or 2 liter per hectare as pre-emergent, 2.5 liter per hectare pendimethalin at 8 days after sowing. Dip seedlings in phosphomidon 0.2% against root nematode. Go for foliar spray with iron sulfate 0.5%, DAP 2%, GA3 3 times at panicle initiation stage. This will give good grain setting. Okay, physiological maturity when ear heads turn yellow, moisture 18 to 20%. Germination standards is 80%. Hybrid seed production in paddy, fertility restorer genes are there. Many restorer genes are there. There is a big list. Go through it. The common examples are RF1 and RF2. Isolation distances for foundation 200 meter and certified 100 meter. Time isolation and barrier isolation is practiced here. 20 days of time isolation and 30 meters tall crops of barrier isolation is practiced. Fertilizer dose is 150, 60 and 60 NPK kg per hectare. To extend pollen supply time, male parent is seeded twice or thrice at 4 to 5 days interval. Row ratio is 8 is to 2, 8 is female and 2 is male, or 10 is to 2. To synchronize the flowering, spray early flowering line with 2% urea or apply 35 kg per hectare urea to delay flowering. For late flowering line, spray 2% DAP to flower early. Draining the field will delay flowering. Giving high stand of water facilitate rapid planicle development. GA3 at the rate of 50 grams per hectare spray increases exertion of the panicles. The third crop is sorghum. Sorghum varietal seed production. Sorghum bicolor is the scientific name. 2n equal to 20 is the chromosome number. 2 spikelets. Spikelet is the panicle name here, or inflorescence is generally called as. One spikelet is sessile, that is bisexual, and one is pedicellate, that is male or sterile. Flowering is basipetal, that is from top to bottom. Isolation distance 200 and 100 for foundation and certified seed. In case of hybrids, it is 300 and 200 for foundation and certified. In Johnson grass, it is 400 and 400 meters distance for foundation and certified seed respectively. Seed rate is 12 kg per hectare. 4H sorghum, it is 400 to 200 isolation distance for the foundation and certified. Seed treatment: soak the seeds in 2% KH2PO4 for 16 hours, 5% carbofuran granules, chlorpyrifos at the rate 4 ml per kg. Then seed dress with bavistine at the rate 2 grams per kg. Azospirillum application at the rate 50 grams per kg are recommended. Number of inspections followed here are 3. Next, recommended days of fertilizer is 100 is to 50 is to 50, that is NPK kg per hectare. Weed control with atrazine, same as maize, at the rate 10 ml per liter as pre-emergent. Major problems are kernel smut and head smut, which are known as designated diseases of sorghum. Germination standards are 75%. 4 male border rows are generally kept for hybrid seed production. Generally, in all cereal crops, if germination falls below 5 to 10% of standards, soak the seeds in double the volume of 10 4m solution, 10 to 14 ml solution of disodium hydrogen phosphate, and dry back to original moisture content of 8 to 10%. Means we should increase the moisture, again we should decrease the moisture, then we can enhance the germination. For hybrid seed production, CGMS system is used. First hybrid is CSH1 released in 1964. Here the male and female rows ratio should be 4:2. First one is female 4 and 2 is the male. Spray malic hydrazide 500 ppm at 45 days after sowing, which delays flowering, or spray CCC 300 ppm to delay the flowering. Presence of B line in A line is pollen shredder, and presence of A line in B line is off type. Remember, presence of B. A is the sterile line and B is the maintainer line. If maintainer is present in sterile means it is pollen shredder. If sterile is present in maintainer means it is off type. Okay. All other conditions, rogging, grading is with the 9/64-inch sieve. Coming to seed production in pearl millet, for varieties Penicetum typhoides. Cross pollination is protogynous type here, means gynoecium matures first, and this is centripetal. Synthetics and composite concept is also popular in pearl millet as it is a potent cross pollinated crop. Synthetics, if more than 5 parental lines are combined with good GCA. Example, CO1 ICMS 7703. Composites 3 to 5 inbreds with no GCA are mixed. Example, WCC 75. Isolation for foundation and certified seed is 400 and 200. Seed rate 18 kg per hectare. Seed treatment, soak with 2% KH2PO4. Metalaxyl at the rate 6 grams per kg. 5% carbofuran 3G granules. Then seed dressing with Bavestin at the rate 2 grams per kg against seed and soil borne diseases. Dose of fertilizer recommendation is 100:50:50 NPK. Then for hybrid seed production, planting ratio for foundation seed it is 4:2 and for certified 6:2. Border rows for foundation seed is 8 and certified is 4. First hybrid in bajra or pearl millet is HB1. Here isolation maintained is 1000 meter and 200 meter for foundation and certified seeds respectively. Jerking is commonly practiced in order to synchronize the flowering. The early formed ear heads of first tillers are pulled out or removed for uniform flowering. It is done at 20 to 25 days after planting or 30 to 40 days, means after flowering 20 to 25 days or 30 to 40 days after sowing. Coming to cotton crop, MH and FW450 are used as gameticides in case of cotton. Here exile presentation is found. Cotton is called as queen of fiber crops. The flower of cotton, if purple spot is seen inner side of petal, it is monoidalphous. Cotton is a often cross-pollinated crop where more than 60% of cross-pollination takes place. Interspecific hybrid Varlakshmi, then DCH32 and Jayalakshmi. Intraspecific is Suguna and Savita. Seed treatment with azospirillum at the rate 600 gram per hectare. Planting ratio 8:2. Isolation distance for foundation 50 and certified 30. Topping is generally practiced, which is nipping the terminal 10 to 12th node of central excessive vegetative growth. Germination standard maintained are 65%. Here 40 ppm of Na or auxin reduces the ball drop. Delinting of seeds with H2SO4 or hydrogen sulfate at the rate of 100 ml per kg of seed for 2 to 3 minutes. Then maturation here is appearance of hairline cracks in balls. Germination is 65%. Cotton dead seeds will float in water. Coming to seed production in sunflower, Helianthus anus is the scientific name, family Asteraceae. Here ray and disc florets are found. Ray florets are generally sterile, sessile, pistillate, whereas the disc florets are bisexual and these are useful. Self incompatibility is generally found. It is having protandry condition. 5 anthers Flowering starts from outer whorl and proceed towards the center. Varietal seed production by open pollination under isolation. General and common popular varieties are CO1, CO2, Morden, K1, K2. Varietal renovation method is Pustovet model. Isolation distance 400 and 200 for foundation and certified seed. Seed rate is 8 to 10 kg per hectare. Pre-sowing seed treatment is generally followed to break the seed dormancy. Soak seeds in 300 ppm ethereal for 8 hours or 0.5% KNO3 for 16 hours. Seed dress with Beviston at the rate 2 grams per kg seeds, that is for removing the pathogens. Seed treatment with Azospirillum at the rate 50 grams per kg of seed. Fertilizer doses 80, 40, 40 NPK kg per hectare. Iron deficiency leads to pollen sterility. Weed control is done with fluchloralin at the rate 2 liter per hectare pre-emergent herbicide. Moisture level at harvest threshing should be 18 to 20%. Germination standard should be 70%. Hybrid seed production BSH1 and KBSH1 are the two potent hybrids here. BSH1 parentage is CMS 234A into RHA 274 and for KBSH1 parents are CMS 234A into D6DI. Planting ratio followed is 4 female and 1 male or 6 female and 1 male. Border rows are generally 2. Germination of hybrids should be 60% and for minimum germination in varieties is 60%. 600 and 400 isolation distance for foundation and certified seed. Coming to seed production in castor. Castor is a cross-pollinated crop. It is having protogyny condition where gynoecium matures first. Rhizome of castor is monoecious, that is pistillate or upper 30 to 50%, staminate at lower part. Pistillate mechanism is very popular and very important for hybrid seed production here. There are three types of pistillate mechanisms: N-type, S-type and NES type. First one is N-type or conventional mechanism. This is governed by recessive sex switching gene maintained by Sib mating. Here progeny segregates into 1:1, male into female. Okay. Then S-type, it is non-conventional mechanism derived from reversals which start out as female and revert to monoecious. That means sex reversal will happen in case of S type. This pistillate mechanism is in detail explained in a separate video and it is available on the channel. You may just go through it. Example for S type variety is VP1. Last is NES system. Here pistillate under moderate temperature produce interspersed staminate flowers under high temperature. 100% pistillate line TSP10R, that is Texas S pistillate 10. First castor hybrid is GCH3. The parentage is TSP10R into JIA 15 by Gujarat. From VP1, that is Gouch, was released. VP1 into VP19 is the parentage of Gouch. Isolation for foundation seed, it is 600 and certified 300. Seed rate generally used is 10 kg per hectare and planting ratio is 3:1 or 4:1 or 6:1. Coming to seed production in tomato. Tomato is a day neutral plant and self-pollinated. Here seed to seed method of seed production is followed. Isolation 50 and 25 for foundation and certified under variety, and for hybrids it is 200 and 100. Seed rate generally 300 to 400 gram per hectare. Grading of tomato seed with 6 by 64 inch sieve. Fertilizer doses all 100 kg per hectare NPK. Planting ratio 10:2 or 12:1. Seeds of tomato are extracted by acid method. Here 10 ml of HCl per kg is used. For hybrid seed production, emasculation and dusting is followed. Coming to seed production in brinjal. Brinjal is Solanum melangena. Here often cross-pollination is found. Method of seed production is again seed to seed. Planting ratio 5:1 to 6:1. Isolation distance 200 meter and 100 meter for foundation and certified. Seed rate 400 to 500 gram per hectare for varieties, whereas 200 gram per hectare female and 50 gram per hectare male is used for hybrids. For F1 seed production, the male and female parent seed rate is 500 grams per hectare. Tomato seeds are graded using 6 by 64 inch sieve. In tomato, hybrid seed production is done by emasculation and hand pollination. Malic hydrazide is used as chemical hybridizing agent. A designated disease of brinjal is little leaf. Four types of flowers are found in brinjal: long style, medium style, short style, and pseudo short style. Genetic purity of brinjal and tomato hybrids is 90%. Coming to seed production in chili, capsicum uh, species, often cross-pollinated, protogynous condition is found. Method of seed production is seed to seed. 400 and 200 meters of isolation distance for foundation and certified seed is practiced. The auxin or NA spray at the rate 4 ml per liter to reduce flower drop. Germination standards are 60%. In bhindi, it is often cross-pollinated. Protogynous condition is found. 400 and 200 isolation distance. 4:1 planting ratio is followed. Hybrid seed production by hand emasculation and pollination. This is just like cotton. Designated diseases are yellow vein mosaic or clearing mosaic virus or hibiscus virus 1. Germination standards are 65%. In case of onion, it is a long day plant. Bulb production is taken here. Day neutral for seed production. Bulb to seed method permits selection of true to type plants in case of onion. Here about 93 to 95% cross pollination is found. 1500 is the isolation distance. Seed to seed or bulb to seed seed production method is followed. Coming to seed production in cucurbits, most cucurbits are monoecious and few are dioecious. Monoecious means male and female found on the same plant, whereas dioecious on the different plants. Okay. So isolation distance 1500 here. Germination minimum 60%. Ethereal 200 to 250 ppm at 2 leaf stage spray increases the female flower numbers. Hybrid seed production, emasculation and hand pollination is practiced for melon seed production. Hand emasculation and insect pollination. GMS for muskmelon is followed, that is genetic male sterility. Use of gynoecious sex forms like in cucumber. By using gynoecious line, muskmelon hybrid Pusa Rasraj was released by IARI. Chemical sex expression: Ethereal 400 ppm, GA3 10 to 25 ppm in cucumber, MH 100 ppm etc. induce female flowers. Remember, Ethereal GA3 MH or malic hydrazide induce the female flowers. Now this was all about seed production in different crops. Coming to seed certification. Seeds of only those varieties notified under Section 5 of Seeds Act 1966 are eligible for certification. Breeder seed is exempted from certification. Foundation and certified class seeds come under certification. Of types. These are plants that differ in morphological characters from rest of the population. Inseparable crop plant seeds like wheat in barley, oats, triticale, rye, gram. All these when it happens, when they are found alike. Okay. Designated diseases in different crops. Designated diseases are the diseases that reduce yield and quality of seeds. In wheat, disease is loose smut and organism is Ustilago triticale. In sorghum, grain smut and head smut by Specialotheca sorghai. Pearl millet, ergot and grain smut, downy mildew. In cowpea, anthracnose by Coletotrichum lindemithianum. In green gram, halo blight by Pseudomonas species. In case of gingerly or sesamum, leaf spot by Cercospora sesami, sunflower, downy mildew, Plasmopara halstead, then in brinjal, Phomopsis blight by Phomopsis vexon, in chili leaf blight by Alternaria solani, and anthracnose by Coletotrichum capsicum. In tomato early blight, leaf spot, tobacco mosaic virus are the designated diseases. The meaning of zoogenesis is one of its kind. For varietal identification, grow-out test and electrophoresis are used. Grow-out test is to determine the genetic purity of seed, whereas electrophoresis is based on protein binding and isoenzyme activity. Coming to seed drying, high moisture content seed with moisture content of more than 17% should be dried first under shade and then sun dried. LSU dryer largely used for paddy. Number of counts to be taken while seed certification and inspection. If the area is less than 5 acres, 5 number of counts are taken. 6 to 10 acres, 6 counts. 10 to 15 acres, 7 counts. 16 to 20 acres, 8 count. 21 to 25 acres means 9 counts are taken. Random positions are fixed and moved in that direction, and the counts are taken. Processing inspection in paddy by flotation, maximum 5% grains floating is permissible. In groundnut, 4% of filled pod are permissible. Uppo first signed in Paris in 1961. Grow test is prerequisite for hybrid seed certification of cotton and castor along with muskmelon and brinjal. Seed Act and rule, I have already discussed about it. The things which we have not discussed till now, I'll just give you here. Seed Act has 25 sections. New varieties evolved by SAUs and ICR institutes are notified and released under Section 5 of Seed Act. Labeling and notified seeds under Section 6A. A, B, C, these things are amendments, means sections were before and they made gradually changes in the subsequent years. So A, B, C, D will be given. That is nothing but amendment. Color, size and content of label are under Section 6B. Example, color of label is opal green and size is 10 cm into 15 cm. Sale of notified varieties under section 7. At present 86 seed testing laboratories are 105 seed testing laboratories are there in country. It may be more also. Appointment of seed analyst is under section 12. Seed inspector appointment is under section 13. Restriction of export and import of seed is under Act 17. Section 17, and central seed lab establishment is under subsection 1 of section 4. Certification agency establishment is under section 8. Grant of certification is under section 9. Central seed committee is section 3. State seed laboratory is under section 4 subsection 2. Term of members of central seed committee is 2 years. Coming to varietal identification, it is done by grow-out test and electrophoresis. Seed drying in paddy, it is LSU dryer. Seed processing to remove awns from barley, that is debearder. Seed treatment for pre-sowing treatment in paddy, 1% KCl for 12 hours. So, soaking is generally practiced. For sorghum, 1% salt or NaCl or 1% KH2PO4 for 12 hours. In pulses, zinc sulfate, magnesium sulfate, then MnSO4 100 ppm for 4 hours. For seed pelleting, adapted in forest tree seeds. Coming to seed storage, this we have already discussed the thumb rule and all, and desiccants, these things already discussed. Coming to packing materials for seed, for moisture and vapor pervious, cloth bags, gunny bags and paper bags are used. Moisture impervious, vapor permeous are polythene bags of less than 300 gauge or urea bags. Moisture and vapor proof are polythene bags of more than 700 gauge, aluminum foils, rigid and plastic bags, etc. Main chemicals used in godowns against pest are malathion 4% dust or 50% EC, phosphine, that is aluminum phosphide. After exposure, they release hydrogen phosphide. Methyl bromide is also used. It is odorless gas, and other gases like chloropicrin are added to detect the leaks. Rodents can be managed by zinc phosphide. Coming to seed marketing, pricing policy. In public sector, prices are based on economic pricing policy. Economic pricing considers effect of seed price on economy. Fumigating in godowns with aluminum phosphide 3 gram per meter cube for the period of 4 to 5 days, and methyl bromide 20 grams per meter cube for the period of 24 to 48 hours are most effective in order to avoid the contaminants like many of the pests and storage pests also. This is all about part 4, friends. If you have any doubt, keep it under comment section. Keep on sharing, subscribing and commenting.